Okay, now I'm going to try to explain meiosis. Here's a cell that is in interphase. It has already duplicated its centrosomes. It's already duplicated its DNA material. So there's 23 chromosomes from your father, double-stranded, 23 double-stranded chromosomes from your mother, present within the nuclear envelope. Now, meiosis 1, it's similar to mitosis, but a lot different. Instead, these chromosomes pair up to form homologous pairs. You have a chromosome from your father, double-stranded, and a double-stranded chromosome from your mother. They share the same genetic material or DNA, and that's what makes them homologous pairs. They line up at the equatorial plate, or equatorial plane. The centrosomes are on each side, opposite sides, and spindle fibers have connected to the centromeres on each side. Now let's take a look at what homologous pair is. Here you have two sister chromatids. This is the father double-stranded chromosome and a mother. They're connected by one centromere here and another centromere here. There's two centromeres. Up here you see there's non-sister chromatids. This is what makes genetic variability happen. And what happens is when they cross over here, where they meet, DNA material is shared at these points. So let's say this is the point strand of DNA that's hair color. It's going to share, and this is what's going to cause genetic variability later on. Here you see they've already lined up, like you've seen here. They've lined up. The spindle fibers have connected to the centromeres, and boom, something sudden happens where they're splitting. And what happens is the double-stranded chromosomes do not split. The centromeres do not split. They just pull the homologous pairs apart to each side, which leaves two genetically different haploid cells. You can see where the crossing over occurs is what causes genetic variability. That is why they're different. Now, if this was happening in the male, these would be two secondary spermatocytes. If this was in the female, you would have one secondary oocyte and your first polar body, which would be a lot smaller than that one. And it will eventually die. Now, here is meiosis 2. In meiosis 2, the double-stranded chromosomes line up again at the equatorial plane by the centrosomes and the spindle fibers connected to the centromeres on both cells. Now, sometimes in the female, your first polar body could already die off, but it might go through meiosis 2 and divide, but it would just come up with two more polar bodies that will die off. In the female, there would only be one, but in the male, both of these are going to divide. Here you see in anaphase 2 of meiosis 2, the centrosomes and the spindle fibers connected to the centromeres are pulling apart, this time, the double-stranded chromosomes, which will leave us with 4 genetically different haploid cells. Now, this is the end of meiosis 2. In the male, we would have 4 spermatids, which will develop into spermatozoa. They'll develop a tail, acrosome, and become a spermatozoa. And in the female, you'll have one large ovum, and you may end up with one primary polar body. You might have a second primary polar body. You might have three. Eventually, these are not going to matter because they're all going to die. They're going to die off. You'll only be left with one ovum. And that's it.